Hi, everyone. Welcome back to the 2025-2026 Oncology Pharmacy Resident Clinical Pearl Series. This is the final video in the 10 video series in which I'll cover the IGNYTE trial that examined the RP1 combined with nivolumab for advanced anti-PD-1 failed melanoma. I find this trial to be pretty interesting because this is a kind of clinical controversy in which there's a lot of speculation as to what led to the failure of the FDA approval of RP1 for advanced or metastatic cutaneous melanoma. For the purpose of this presentation, I'll mainly focus on the clinical components of it rather than diving deeper into the politics of why there may have been a failure of the drug being approved by the FDA. For the objectives of this presentation, I will introduce the background of the IGNYTE trial, and this is specifically on the phase 2 trial because the phase 2 trial has all of the results released, while the phase 3 trial has not released all the results at the time of this recording, presumably because of the failure of the RP1 drug being approved by the FDA. After going over the actual phase 2 trial, I will then evaluate the FDA's stance on the regulatory approval of RP1. Now, to begin with the background of the treatment of unresectable metastatic cutaneous melanoma, the NCCN guidelines does recommend the use of immune checkpoint inhibitors for frontline treatment. The limitation of this is that a majority of patients will have innate resistance to single-agent PD-1 inhibitors. Now, you may be thinking, okay, but the NCCN guidelines does prefer the use of ipilimumab and nivolumab. However, the limitation with this combination is that 40% of patients will have innate resistance to that, and a quarter of patients will acquire resistance to PD-1 inhibitors within 2 years of treatment. Despite the fact that a majority of patients will have innate resistance to PD-1 inhibitors, or potentially acquire them after 2-year treatment, the DREAM sequence trial did show that in treatment-naive BRAF V600E mutant metastatic melanoma patients, that the use of immune checkpoint inhibitor combination would have a statistically significant difference in better overall survival and median duration of response compared to targeted therapies for BRAF V600E. Shown on this slide is the current recommendations for treating metastatic or unresectable cutaneous melanoma, which shows that the NCCN guideline does prefer the use of immune checkpoint inhibitors over more targeted therapies at this time. Another treatment option that can be considered for the treatment of unresectable metastatic cutaneous melanoma in adult patients is lifileucel. This is the only FDA-approved tumor-infiltrating lymphocyte therapy that was validated from the C14401 study that found that it would have an objective response rate of around 31%, median overall survival around 14 months, median progression-free survival of 4 months, median duration of response not reached at a median follow-up of around 28 months, with 42% of the response maintained at least 18 months. However, the issue with this drug was that the percentage of more severe adverse events was found in the majority of patients with thrombocytopenia and anemia, and then there were around 42% that experienced fibrotic neutropenia. So based on the results of this study and the fact that patients could develop acquired resistance PD-1 inhibitors, and a lot of patients would also have innate resistance to immune checkpoint inhibitors, the investigators of this IGNYTE trial were seeking to see if RP1 could provide both better efficacy outcomes and better toleration with lower adverse events. As previously mentioned, the study objective of this phase 2 clinical trial was to evaluate the impact of RP1 in combination with nivolumab on clinical efficacy outcomes in patients with unresectable stage 3B or 4 cutaneous melanoma with confirmed disease progression after treatment with an anti-PD-1 containing treatment regimen. The IGNYTE trial is a open-label multi-center phase 2 trial that was funded by Replimune, which was also the manufacturer of RP1. The patients that were included in this trial were adult patients with unresectable stage 3B or 4 cutaneous melanoma who had disease progression while being treated with anti-PD-1 monotherapy or in combination with another anti-cancer agent, including the adjuvant setting. They excluded patients who had a history of serious complications from immune checkpoint inhibitor therapy to avoid any unnecessary harm from the use of nivolumab. They also excluded patients who were on current antiviral therapy or prior oncolytic therapy to not interfere with the efficacy of RP1, which is a herpes simplex virus-based oncolytic immunotherapy. Shown on this slide is the intervention that was used for the IGNYTE trial. For outcome measures, the primary endpoint was overall response rate, while the secondary endpoints were duration of response, complete response rate, progression-free survival, and overall survival after 1 and 2 years. Regarding the statistical analysis of the IGNYTE trial, the investigators determined that a sample size of 125 patients would provide greater than 97% power to reject the null hypothesis of an overall response rate less than 15%. The Kaplan-Meier method was used to estimate the duration of response, progression-free response, and overall survival. Shown on this slide are the notable baseline characteristics. As you can see on this slide, the majority of patients did have a wild-type BRAF status, PD-L1 negative status, prior therapy with anti-PD-1 other than as adjuvant therapy. There was also 65.7% of patients who had primary resistance to prior anti-PD-1, and then there were around 34% of patients that had secondary resistance to prior anti-PD-1. This will be important to consider when I do give my thoughts on the study's limitations and whether or not the FDA made the right call in not approving this drug. The investigators ultimately found that with this treatment combination, there was an objective response rate of around 33%. The median duration response was around 34 months. The median progression-free survival was around 4 months. The median overall survival was not reached. And when it came to the overall survival rate after a year, it was around 75%, while the 2-year overall survival rate is around 63%. For the drug-related adverse events that were experienced by patients in this trial, 90% did experience adverse events of any grade, but there was a very low percentage of severe adverse events in which around 13% of patients experienced grade 3 or 4 events, while no patients experienced grade 5 events. The most common adverse event of any grade that was experienced by patients was fatigue. Ultimately, the investigators of the IGNYTE trial concluded that RP1 combined with nivolumab demonstrated clinically meaningful and durable responses with a favorable safety profile. To start with the strength of the trial, the baseline characteristics of patients enrolled are most representative of patients with cutaneous melanoma. They also did have subgroup analysis that showed how beneficial this novel treatment could be for the selected patient population, depending on characteristics such as the type of immune checkpoint inhibitors that they previously received, the setting in which the patients were treated in, such as neoadjuvant or adjuvant settings, the degree of PD-L1 expressing tumors, the stage of disease, and BRAF mutation status. Despite these strengths, there were notable limitations. One was that since this is a phase 2 trial, they did not directly compare RP1 nivolumab with other recommended second-line or subsequent treatment options for unresectable stage 3 or 4 melanoma. The other genetic mutations such as ROS1, KIT, and NTRK1, 2, and 3 were also not examined, and this would have been helpful to further delineate how efficacious this treatment could be. It can also be argued that with the fact that these patients failed the immune checkpoint inhibitor, adding on nivolumab with RP1 would also not be particularly helpful for the treatment of this patient population, because if a patient were to still have disease progression after immune checkpoint inhibitor therapy, adding it on along with another novel treatment would also likely not be expected to have a significant impact on the treatment of the patient's disease. Ultimately, the FDA did deny approving RP1 in combination with nivolumab due to concerns that the phase 2 IGNYTE trial did not have a well-controlled study. There was an inability to isolate RP1's clinical benefit when using combination with nivolumab, and that its heterogeneous population would hinder interpretability. To break down these reasonings, to start with the first about the well-controlled study component, this is a bit questionable because the investigators did determine that a patient population of 125 would provide a study of over 97% power. So unless the investigators' determination of this was not done accurately, I would not say this is a valid concern. For a second reason of the inability to isolate the clinical benefit, since patients did have prior disease progression with an immune checkpoint inhibitor, theoretically you could say that nivolumab at that point would not be helpful for treating the disease at all. So if the patient does have any degree of response, then it would be solely based on RP1. But at that point, the question would be why the investigators would use nivolumab in the first place if the patient did have disease progression with an immune checkpoint inhibitor. So this really is a flawed design where they could have used other targeted therapies based on a patient's mutational status to see if there could have been additional benefit with that combination. It's also limited just solely based on the fact that this was a phase 2 study, so there's not comparisons with other comparator arms that were based on standard of care treatments. Lastly, with the heterogeneous population aspect, they did examine patients who previously were treated with immune checkpoint inhibitors in the adjuvant setting or with other BRAF targeted therapies. This is where the approval based on this broad population could be difficult to determine, especially since there was not a comparison with other standard of care treatments. So based on these reasonings overall, the FDA denying the approval is reasonable because the lack of comparison with other recommended treatments really calls into question how efficacious RP1 could be compared to standard of care treatments. And this is where it gets complicated because you could argue that in other clinical trials there have been cases where the clinical comparisons of the study drug and the control group may not necessarily be as good of a clinical comparison. But in this case, I would say that based on what NCCN recommends and what could be beneficial for the patient, it's really questionable as to how beneficial RP1 could be based on what was examined in this phase 2 trial. These are the references that I used for this presentation. Feel free to comment about your thoughts on the IGNYTE trial and whether you think the FDA was ultimately justified in denying the approval of RP1 in combination with nivolumab for the treatment of metastatic or unresectable melanoma. If you do have any questions or want to provide feedback directly to me, my Monument Health email is cchen1@monument.health. Thank you.